Welcome to the pod Biomanufacturing and Fermentation Technology. In this episode, we translate microbial science, process engineering, and manufacturing realities into insights that scale.
Welcome back to the deep dive. If you're in biotech, you probably know this story. It's a gut-wrenching one. You spend months, maybe years, in the lab perfecting a process. You get these fantastic yields from your little 5-liter benchtop fermenter. Your organism is happy. You think you've cracked it.
And then comes the big moment.
Then you hit the scale button. You go to 10,000 liters, 50,000 liters, and PFFT. That beautiful performance just vanishes.
It's costly, it's unexpected, and it's where, really, biological potential meets industrial reality. It also exposes the limits of what we'd call traditional scale-up.
Right, and that's why we're seeing this shift, this move towards a different framework altogether.
Yeah.
Scale translation.
Exactly.
Okay, so let's unpack that right away, because traditionally scale-up seemed pretty simple, right? The goal is just to preserve a few key engineering parameters.
That's it. You'd aim for, say, a constant power per volume, or P over V, basically the amount of energy you're putting into the fluid.
Or maybe a constant CLA, the oxygen mass transfer coefficient.
Right. And the logic was, if you keep those averages constant, the biology should just follow along.
But that assumption is, it's fundamentally flawed at large scales.
It is. Scale translation starts from a different place. It recognizes that you actually can't keep all those parameters constant at the same time. It's physically impossible. When a vessel gets 100 times taller, the whole environment changes.
So if we can't just keep the averages the same, what is the new goalpost? What does success even look like?
The key idea, the translation principle, is that success is defined by reproducing the cell exposure distribution.
Okay, cell exposure distribution. What does that mean in practice?
It means we're moving past the averages and focusing on the microbe's actual experience.
Yeah.
What matters is the amplitude and the frequency of stress, those quick drops in dissolved oxygen or pH or substrate, and crucially, how long the cells are stuck in those bad zones.
That feels like a huge paradigm shift. And if the average set points are misleading, what does that say about the role of the pilot scale? Because that's always been seen as the final proof.
Well, the sources are really clear on this. The pilot scale's main job is not performance. It's a risk exposure platform.
A risk exposure platform.
Its job is to show you the mismatch between your perfect, homogeneous lab assumptions and the messy reality of manufacturing, with all its mixing limits and sensor lags.
So if you run a pilot and it's too clean, you've actually learned nothing.
You've gained false confidence. You haven't stressed the system enough to find out where it's going to break at full scale.
Let's get into the physics of why that happens.
Yeah.
Because this is really where the money gets lost. As vessels get bigger, the flow just isn't uniform, and the mixing time, it skyrockets.
Yes, and that creates these sustained invisible gradients in everything the cell needs to survive.
Which brings us directly to a really key concept, the bulk DO fallacy.
Yes, the big one.
So tell me about that. Why is that bulk reading on the screen lying to the operator?
Well, the bulk DO fallacy is this simple reality. Just because your one main probe says you have acceptable dissolved oxygen, that doesn't mean oxygen is sufficient everywhere.
Right.
In a large, tall vessel or in a thick, viscous broth, you get these incredibly steep gradients. The time it takes for oxygen to get from the sparger at the bottom all the way up and into the cell mass, the OTR is just severely limited in certain areas.
So the operator sees a green light, maybe 30% saturation, feeling good, but way down at the bottom or far from the impeller, the cells are literally starving. The local concentration is near zero.
Exactly. And the consequence of that cycling, the cell getting shuttled between a good zone and a bad zone, is devastating. Even if the average DO is fine, these oscillations trigger stress responses, what we call overflow metabolism.
So the cell just stops making your product.
It panics. It stops focusing on your product and starts making waste products like acetate. Your yield just plummets.
There's a perfect example of this in the source material with Bacillus ligeniformis, the workhorse for making industrial enzymes.
A classic case. In the lab, in a 5-liter vessel, everything's uniform. You get great yields, over 10 grams per liter of protease.
But then you go big, 50,000, maybe 100,000 liters.
And the physics takes over. Mixing times jump from milliseconds to maybe 30, even 60 seconds. And that creates these huge vertical DO gradients that swing from, say, 50% saturation at the top to basically zero at the bottom.
So the cell is on a 60-second roller coaster ride from feast to famine.
Precisely. And that cycling through these hypoxic, these low oxygen zones, forces it into that inefficient state. It starts pumping out acetate.
Yeah, the business impact.
A 20 to 30% cut in enzyme yields. If that's a multi-million-dollar product, I mean, that's just catastrophic. It shows why you have to treat mixing and gradients as primary biological factors.
Okay, so that leads us to the engineering solution. If we know we can't hold everything constant, how do we actually manage the scale change? We use tools like dimensionless numbers, but differently, with selective similarity.
Selective similarity is all about making smart trade-offs. Dimensionless numbers are great tools, the Reynolds number for turbulence, PV, Kli, all of them. But just picking one and trying to hold it constant is, well, it's scientifically inadequate.
So the question shifts from which parameter do I keep constant to something else.
2, which deviations will push my organism over its breaking point? That's the pivot.
So you prioritize based on the organism's known weaknesses.
Exactly. For a highly aerobic process, you might prioritize a high OTR margin above all else. For something that produces a lot of acid, you might prioritize minimizing mixing time to kill pH gradients. Those are your prioritized criteria.
And you might let other things slide, like, say, exact geometric similarity.
You might have to, because that often conflicts with what the manufacturing plant can actually do. You deprioritize things that aren't the primary physiological bottleneck.
Is there a concrete example of this kind of prioritization?
Sure, think about antibiotic production with Streptomyces claviligeralis. This bug is extremely sensitive to pockets of high glucose.
Right, if you feed it too fast in one spot.
At large scale, if your mixing is poor, the glucose feed creates these intense local pulses, and that pushes the organism into acid-producing pathways, and your yields can drop by 15, even 25%.
So what do they prioritize to solve that?
They prioritize maintaining a very high Reynolds number, something over 100,000. This drives the choice of the impeller and the speed, all to ensure really intense circulation. They're focusing on bulk liquid movement to solve the substrate gradient problem, which is the organism's known weak spot.
Even if that means maybe compromising a little bit on something else, like the maximum possible killer.
Exactly. You sacrifice a little on one front to solve the problem that will actually kill your process.
Okay. But if the whole point of a pilot run is to find these vulnerabilities, and we know they're often too clean to do that, how do you proactively find that breaking point? You have to intentionally create the stress.
And that is the entire purpose of stress emulation. You figure out what those large-scale gradients will look like, the frequency, the amplitude, and you deliberately reproduce them in a controlled lab-scale system.
So you're making the lab intentionally messy, but in a very precise way.
Yeah.
How do you even do that? How do you mimic a 100,000-liter tank on a bench?
You use what are called scaled-down models. Sometimes it's a single vessel where you just program the controller to oscillate the conditions. Or you can get more complex with multi-compartment models.
What does that look like?
You might have a 2-compartment reactor, a small, well-mixed, highly aerated compartment, which is like the top of the big tank, and it's connected by pumps to a large, poorly mixed, low aeration bottom compartment.
And the cells get pumped between them.
At a flow rate that's set to match the calculated mixing time of the full-scale vessel, they physically recreate that stress cycle.
And the goal of these emulation tests isn't to get the best performance.
No, not at all. The goal is to define the failure boundaries. You're looking for the exact point where productivity just falls off a cliff, or where your impurity profile suddenly gets much worse.
And those boundaries become your manufacturing specs.
They become rigid manufacturing specs. For instance, acetate formation skyrockets if exposure to DO below 5% lasts longer than 45 seconds. That now defines the absolute maximum mixing time your plant has to achieve.
So going back to the Streptomyces example, you'd be introducing controlled glucose pulses.
Yep, maybe 5 to 10 grams per liter every 10 or 20 minutes to map the mixing time and see exactly where that byproduct formation takes off.
You're defining the organism's tolerance to the industrial reality.
Precisely. But there's another layer here, the big reality check. Facility constraints. Because even if your pilot works, if the production plant can't physically do what the pilot plant did, you're still going to fail.
This is where that false confidence really becomes dangerous. The pilot vessel probably has way more power and better cooling than the huge production tank.
It's a massive translation gap. When you scale up, volume increases by the cube, but the surface area for heat transfer only increases by the square.
That ratio mismatch is brutal.
It means the production vessel has way less ability to remove all the heat the metabolism is generating. Very often, that heat removal is the true limiting factor at industrial scale.
It's a hard physical ceiling.
Absolutely. So you have to align your testing with what manufacturing is actually capable of. You check the actuation limits, what's the plant's max cooling duty, what's its max aeration rate, and you have to account for utility variability. Cooling water temps can drift in a way they just don't in a pristine pilot facility.
And then there's the whole instrumentation gap. I was really struck by that detail in the source material about how a sensor itself can lie to you in a thick broth.
Oh, yes. In very viscous fermentations, like some yeast processes, the broth can get over 50 centipoise. It's thick. And a stagnant liquid film can actually form right on the surface of your dissolved oxygen probe.
So the probe isn't even seeing what the cells are seeing in real time.
It can't. The film physically slows down oxygen diffusion to the sensor. It's a measurement artifact. And it means your DO reading actually becomes dependent on your stirrer speed. Speed it up, you strip the film, the reading changes.
That leads to false oxygen security.
The worst kind. You think you're safe, but the sensor is just too slow to catch the brief, lethal DO dips that are killing your cells.
That is just a conspiracy of biology, engineering, and physics all working against you. So if these failures are so insidious, what are the warning signs? What are the diagnostic signatures you can spot before the batch collapses?
These are so critical because they help you tell the difference between an engineering problem, like a gradient, and a simple biological problem.
Okay, let's start with local oxygen limitation. We know it causes overflow metabolism. How do you spot the invisible gradient on your control screen?
The key signature is this. Your DO controller output starts climbing dramatically. It's demanding more and more air, more and more agitation, but your bulk DO reading stays perfectly stable and acceptable.
So the controller is screaming for help.
It's screaming. That rising output is telling you there's a huge localized oxygen demand the system is struggling to meet, even though the single probe looks fine.
Okay, what about mixing limited substrate gradients? The high glucose pockets.
There you see a really nonlinear link between your feed rate and your oxygen uptake rate, or your co-evolution. The metabolism isn't smooth and predictable, it's spiky. And the batch becomes incredibly sensitive to tiny changes in where you put your feed pipe.
And finally, that instrumentation artifact, the slow DO probe.
The diagnostic is to test it. If the probe response gets really sluggish, and especially if the reading itself changes when you just change the stirrer speed, you've got false security from that surface film.
And these problems are just magnified in something like, say, aspergillus niger for citric acid production.
Oh, absolutely. Those broths can hit 100 centipoise. They're like sludge. The poor mixing creates these localized pH gradients from the acid, and those pH dips can cause the fungus to sporulate. You can lose 20 to 40% of your yield just from that.
So the solution has to be rigid.
It has to be. You prioritize an aggressive mixing time threshold, maybe under 30 seconds, and you make absolutely sure the plant can hit that target without overheating.
So what does this all mean for you listening? Scale translation is really this reconciliation of what's biologically possible with what's industrially real. We have to stop managing averages and start managing the actual cellular experience, which is all about those gradients.
That's the core takeaway. A strong strategy uses selective criteria. It focuses intensely on the things that are truly limiting for that organism OTR margins, co-Eurostripping, critical mixing times. Success at scale really does hinge on anticipating the physiological impact of physical forces.
Which brings us to a final thought for you to think about. If the best scaling criteria change depending on the product, say you shift from needing high OTR for an enzyme to needing high co-Eurostripping for bioethanol, how would the minimum data set you collect in your early lab work need to change?
Right. What new biological thresholds would you have to define right from the beginning to make sure you're even asking the right questions for scale-up?
What data are you currently leaving on the table? We've reached the end of this deep dive. We'll see you next time.
You've been listening to Biomanufacturing and Fermentation Technology with Prasad Urnala. Thank you for joining me. Until next time, keep engineering biology into reliable manufacturing.